Sadia, please unmute yourself. Okay. So out of 20 experiments, students, all 20 experiments, there is no need to perform. Okay. There are some demo experiments. Like there are some, like the next experiment, that is experiment number 5. It is the study of abiotic and biotic factors in aquatic ecosystem. Okay. So aquatic ecosystem means you have to go underground water. And you have to go underwater and do, it's very difficult, it's not possible. So those kind of experiments where it is not possible to calculate about the ecosystem, to calculate the frequency and abundance of the ecosystem, like for example, just now I said aquatic ecosystem, we'll consider it to be as a demo experiment. Okay, wherein we are not performing it, but you will be writing it in your record book. I am repeating, out of 20 experiments, there are some experiments which are not possible to perform. Okay, like for example, the next experiment, experiment number 5, it is about aquatic ecosystem, wherein you have to go underwater, underneath the water, and see the light intensity, pH, and all those things. It's highly impossible. So those type of experiments which we are not able to perform are considered to be as a demo experiment. We'll not do them, but we will mention it in our record books. Clear?
Okay.
Yes, clear, ma'am.
Yes, ma'am.
One question in that case, what do we have to do with the tabulation in the record?
Okay, leave it empty. If any readings are given from the university, we'll mention those readings. Otherwise, your records will be certified if those experiments are left empty also, because it is understood. Okay, if I am the one who is yes, somebody, Netra is telling, yes, yes, Netra. When we have to submit the record book, that's what I said. When Dikshita asked, if you are coming to college next week, come with the written experiments. Otherwise, whenever you are coming to college, how many ever experiments are completed, so many experiments should be written in your record book.
Ma'am, thank you, ma'am.
Will write and come from the house only, ma'am? From our...
Yeah, write and come. That's what I said in the last class. That is yesterday's class, I told you. When you are submitting, it should be written in your record book, except for the calculations. Calculations, that is the readings, we will do the experiment and we'll put those experiment in the record. That's all.
Ma'am, whatever is in this manual.
Excuse me, madam.
Yes.
Experiment we have explained, that much is enough for next week.
Yeah.
For books.
Like I am finishing 5 experiments today. I'll try to do 5 experiments. Otherwise, 4 experiments should be written in the record book. Must still should be written in the record book.
Okay, madam. What about manual, madam?
Manual is for your reference to write it properly.
Okay.
We are actually following the manual method only. I am following the manual method only. You don't have to mention anything in the manual. Manual is to write the record book. Like principle, we did not do principle during our classes. We have to copy the principle in the record. Proper procedure should be copied in the record. That is why we are using manual.
Only record book we have to submit for you, no, ma'am?
Yeah. Record book should be submitted to me.
Okay, thank you.
Yes, Raushani, what question you have?
Ma'am, I just wanted to ask about this record book. So, like this practical book should have one side plain, left side plain, or left side graph, because some of the experiments also, includes graph, like our first experiment regarding the quadrant.
Yeah.
So that's the confusion. Or should we carry the loose graph papers, and wherever it is required, we should just attach it to the staple it how it is?
Yes, that's what I told you in the yesterday's class, in the first class itself. You will be having a record book which will be having one plain side and one rule side, and then you will be having loose graph sheets. Whichever experiment demands to plot the graph, you will plot the graph and you will staple the graph in that particular experiment.
Okay. All right. Thank you, ma'am.
Yeah. Okay. So shall we start with the experiment now? Experiment number 4. This is a very easy experiment. Okay. The name of the experiment is determination of standing crop Please make a note of it. Determination of standing crop and biomass in terrestrial ecosystem. Somebody has unmute, please mute. Okay. So the name of the experiment is determination of standing crop and biomass in the terrestrial ecosystem. So here you are getting three different terminologies in the name of the experiment. One is you have something called as standing crop. The other terminology is biomass. And the third terminology, what you are getting in this particular heading is terrestrial ecosystem. Okay, let us take it as four. One is terrestrial, the other is ecosystem. All right. So we should understand what are these terminologies. Terrestrial means it is clearly understood those organisms which are present on the land, on soil, on the ground, above the ground. So those organisms which are present on the land, on soil, whatever you consider soil, land, above the ground, however you call it as, organisms present on land in general are called as terrestrial organisms, and organisms which are present underwater are called as aquatic organisms. This is probably a third standard difference. Third standard students will also be knowing what is the difference between terrestrial and what is the meaning of terrestrial organisms and what is the meaning of aquatic organisms. Clear? So now we know that organisms which are present on the ground are called as terrestrial organisms, and organisms which are present on the underwater are called as aquatic organisms. Okay, so the meaning of terrestrial we know. Terrestrial organisms are land organisms. Land organisms are called as terrestrial organisms. Next terminology, ecosystem. What is the meaning of ecosystem? Okay, let us understand the meaning of ecosystem. Ecosystem is consisting of living organisms. All the living organisms, simple living organisms, complex living organisms, all the living organisms present in one particular area. Okay, if we take man is a complex organism. Then if we take plants which are on the land are a complex organism. Then there are simple non-vascular plants present. Then you have microorganisms present. Then you have simple animals and complex animals present. So many organisms living in one particular area, in one particular unit is called as ecosystem. The very easy example for ecosystem to be understood is a pond. In a pond, you have tadpoles present, you have fishes present, you have grown-up frogs present. That is, after metamorphosis, what frogs you get? Adult frogs. Frogs present, you have some microbes present, you have zooplankton, you have aquatic plants, then you have so many things present in a pond. Okay, probably above the limit, we don't know, so many microbes are present. In those microbes you have wide variety of microorganisms. You have algae present, you have this fungi like aspergillus present. So many organisms are present in a pond. So pond is a type of ecosystem where you have simple as well as complex organisms living together. Okay, so that is called as ecosystem. So what is ecosystem? It is a unit area where all simple and complex organisms live. So we know the meaning of terrestrial organisms and and ecosystem. So the full definition for terrestrial ecosystem means you are only concentrating on land ecosystem. we are not concentrating on aquatic ecosystem. Okay, which means we are determining the standing crop and biomass of land ecosystem, organisms, land of land and ecosystem of land, terrestrial organisms present on the land. So this is what we are concentrating here. So in this particular experiment, our main focus is on land, be it a flora or a fauna, but we are concentrating on land. Whereas in the fifth experiment, we'll be concentrating into aquatic ecosystem, that is underneath or underwater ecosystem. Clear? Now next definition is what is the meaning of standing crop. Okay, before standing crop, let me tell you what is the meaning of biomass. Biomass is the total weight or total quantity of organisms in that particular region. Okay, it can be one animal. Like for example, if I take this particular region and here we are only concentrating on dogs. How many dogs are present individually? What is the quantity of dogs in this particular region? Which means we are considering the biomass of dogs. Clear? If we are only concentrating on this region and here I am only looking for cats, how many cats are present? So we are calculating the biomass of cats. So biomass means it is the total weight or total quantity of organisms. Organism can be a fauna, organism can be a flora, or we can together take the organisms. Okay, I can individually take dogs or I can individually take cats as the biomass, or I will also take together dogs and cats together in this particular region. Clear? Biomass means it is the total weight or total quantity of organisms. Organisms can be of flora, organisms can be fauna, individual flora, individual fauna, or organisms can together be complex organisms. Together also we'll be considering how many in number, how many dogs, how many cats, how many plants, how many sheep, how many hens in that particular region is called as biomass. Is the definition clear? What about biomass, ecosystem and terrestrial organisms?
Yes, ma'am.
Okay, so please make a note of it. Terrestrial organisms means land organisms. Ecosystem means it is the unit area where the organisms are living together, be it a simple organism or a complex organism. And biomass means it is the total quantity or total weight of organisms. Organisms can be single organisms, single flora, single fauna, or together we can take the biomass of that particular region. Okay. Please write the definition. If it is done, let me know.
It's done, ma'am.
Now we are coming to the next terminology, standing crop. Standing crop means it is the weight for that particular movement. You are considering the standing crop for that particular movement. Now, when I am taking a biomass of dogs in this particular region, here the weight, the individuality, the biomass can be probably 50. Later, the dogs can migrate to some other region or the sheep can migrate to some other region. So standing crop means at that particular time, at that particular movement, whatever I will calculate, that is called as standing crop. So the definition for standing crop can be given as the weight or quantity of organism in an area at a movement. At a moment. Not movement, it is moment. Okay. So the weight or quantity of organisms in a particular area at a moment. At a moment, at a particular time, what is the weight or what is the quantity of organism? That is called as standing crop. If it is done, please let me know.
Yes, ma'am. Yes, ma'am.
Okay.
Yes,
So I have given you the definition of standing crop. I have given you the definition of biomass. I have given you the definition of terrestrial ecosystem. Okay. Now, what are we supposed to calculate here? So we very well know that we are determining the standing crop of a biomass. For the determination of the standing crop of a biomass, for any experiment, we have something called as requirements. Other branches of science, like in biology or in physics or chemistry, these requirements are termed as apparatus. Okay, since it is environmental science and ecology, we are usually telling them as requirements, something which is required to perform the experiment. Okay, so what are the requirements needed? The requirements which are needed for this particular experiment is, we want a quadrat. We want a specific size quadrat, an appropriate size quadrat. So you know what is a quadrat by now? Quadrat, what is the appropriate size of quadrat which I gave you yesterday?
100 cross 100.
So 100 into 100 square centimeters was a appropriate size of quadrat. And to make a quadrat, what all you require?
Nail, thread.
Nail, thread.
Nails you require threads. Okay, so you know how to make a quadrat now. All right. So we want okay. So we want a quadrat first. Then we want a plover. All of you know what is a plover, a weeding instrument. Okay. And then we want a digger. Okay. And then we want polythene bags. Then I want an oven. And then digital weighing balance. These are the things which are required for the experiment. Okay. We want a quadrat. We want a plover. We want a digger. A plover, we want a digger, we want polythene bag, oven, and digital weighing balance. I hope all of you have seen plover, digger, polythene bags, oven. Oven can be a microwave also. Okay, we are only drawing here. Then digital weighing balance. Digital weighing balance also, I hope all of you have seen. Quadrat we have to construct. Okay, so these are the requirements which are needed to perform the experiment. Please make a note in your books. Once it is done, let us move further.
Yes, ma'am.
Okay, and this experiment you can perform in your fields only. You don't have to come and do it in the college. You can individually get your readings. It's a very easy experiment. If you know, once I tell you the procedure, if you have all these things, if you can do it, you do in your fields. Okay, since yesterday some of them said you cannot come to Bangalore, and Bangalore has a regional center of IGNOU and all those things. If you are staying far away from Bangalore, you can do in your own fields. You can, if you are in, especially those people who are staying near these field areas. Okay, so there you can do it. It is very easy to perform. You have to only take the dry weight and fresh weight. That's all. Okay, the dry weight is the fresh weight what you will be taking. If oven is there with you, and all the other things will definitely be there in every house. So, and with digital weighing balance is required. So you can do it in your places only and note down the readings. Okay, you don't have to come down here only to the regional center and do it. If those things are possible, if those things are available with you, you can do it in your places itself. Clear?
Yes, ma'am.
Okay. Now, how are we supposed to perform this experiment? Procedure required is, first what we'll do is, we'll take an appropriate quadrat. Okay, we have a very huge field. So in a very huge field we cannot do, so we are taking quadrats. All right. So we'll take an appropriate quadrat. So appropriate quadrat in your books, they must have mentioned 50 into 50. You can take 50 into 50 also. You can take 100 into 100 quadrat also. Since I taught you 100 into 100 yesterday, I am considering it as 100 into 100. or else you can take an appropriate size of 50 into 50 quadrat. So first we'll make a quadrat, okay? So we are making a 50 into 50 quadrat. So you have 10, 20, 30, 40 and 50. Okay, depends. You can take 100 into 100 also. If you want to cover a larger area, you can take the 100 into 100 also, or 50 into 50 is also fine since your books are mentioned. So we are taking the 50 into 50 quadrat, okay? After taking this much of area, 50 into 50 or 100 into 100 quadrat area, what we'll do is we will weed out. I'm writing the spelling here, weed out. Weed out means remove. Okay, so quadrat means first you are taking this much of place, or this much of place you are taking, you are considering it. This is just study area right now. Okay, if this is my study area, whatever is present in this particular study area, I am removing it all. I am weeding it all. How will I weed? If there are small rocks or small stones present, I can manually pick them up. If there are some leaves or some plants present, I will be using a plower to remove them all. That is where we are using plower, which we have written in the requirement. Okay, plowers are used to remove, weed out all the things present in that particular area. Okay, this is my growing limit, and in this growing limit, what I'm doing, whatever is present, I am first considering my quadrat, and this is my study material, this is my study area. In this study area, whatever things I have, be it a litter, okay, be it plants, be it stones, be it paper, plastic, whatever it is, some earthworms, okay, some larvae, okay, and then whatever you find in that particular area, you'll have some ants, so all insects will be present. So all these things, whatever is there, you are removing them all. Clear? You will remove them all, and where will you put? You will remove them all and put up in a dustbin or throw somewhere? No, you are not throwing them. You will remove them all, and you will collect them all in a polythene bag. Okay, we are weeding out everything, we are removing everything, and we are putting them in a polythene bag. All right, and after I put everything in polythene bag, I will label that particular polythene bag as polythene bag A. Okay, I am collecting everything together, and I am taking everything in a polythene bag, and I will write the name of the polythene bag as polythene bag A. All right. What I'll do is, after taking the polythene, everything into polythene bag, after collecting everything into polythene bag, I will now take the weight. Let us take the weight of it. Okay. Let us take the weight of it. Whatever is the weight, together weight will take. We'll take a together weight. After together weight of what? I'll just collect everything, put it on the weighing balance, and I will check the weight. I will note down the weight. Then what I'll do is, I'll collect all the leaves and the plant parts. Okay, in my, or else what I'll do, let us do one thing. Let us not weigh here. Okay, please scratch it out, weigh. So you will have stones, you will have plastic, you will have some spoons, you will have some paper, some littering. Everything will come into this polythene bag, and we are naming that litter as polythene bag A. Okay, and then only we'll collect all the leaves and all the plant parts, plant material, and we'll put them into a second polythene bag, and we'll name the polythene bag as polythene bag B. So in first polythene bag, I have soil, I have sand, I have stone, I have paper, I have plastic, I have cups, I have this and that, which is not a plant material, an inorganic material. Whereas in the second polythene bag, I'll be having only the plant material. Okay, I am only taking plant material in the second polythene bag. And in third polythene bag, what I'll do is I'll take the animal material. Animal material, like as I said you, ants can be there in your place. Larvae will be there, some butterfly larva or any larva types of larva. And then you will be having some insects present. Some earthworms will be there. Okay. So all these will be going, all these organisms. Rajani, please mute yourself. So all those animal material will be going in my third polythene bag, and the third polythene bag is labeled as polythene bag C. I am repeating again, students. I'll be taking a study area, an appropriate quadrat of 50 into 50 or 100 into 100 as per your wish. After considering that particular area, what we'll do is we'll weed out everything from that area with the help of a plower. If stones are there, plastic is there in the particular place, some papers are fallen down, some litter is there, everything we'll collect and we'll put up into a polythene bag and we'll label that polythene bag as polythene bag A. Then we'll collect all the leaf and the plant material from that area only, and we'll put all the leaf and plant material into another polythene bag, and we are labeling the polythene bag as polythene bag B. Then we are taking all the animal material, like animals like ants, larva, earthworm, insects, all such things, and we are putting them into a third polythene bag, and we are naming the polythene bag as polythene bag C. So 3 polythene bags you have done without digging. Only by weeding, only by removing, you have created 3 polythene bags. When the study area is completely clean, now what I will do is, I will take a digger and then I'll start digging the study area. I will dig from 20 to 25 centimeter deep. Okay, I am digging 20 to 25 centimeter deep, and I will take off all the materials from there, underground parts. So when I am digging, I'll get some plants and some other things. Okay, plant material inside the ground. First, in my second polythene bag, I had the plant material above the ground. Now in my fourth polythene bag, I am having the plant material which is present inside the ground. I am digging and I am collecting all the materials which are present, especially the plant material which is present inside the ground, and I will create another polythene bag as polythene bag 4, and I will name it as polythene bag D. Did you understand till here?
Yes, ma'am.
Yes.
Before digging, we have made 3 polythene bags. After digging, we are making 1 polythene bag. Clear? So totally we have 4 polythene bags.
Ma'am,
Okay. Yes.
in this third bag, fourth bag, D,
Yeah.
plant material and animal matter is also there. Animal part is also there.
Yeah. If it is underground and if you are getting it, you will take that. Okay.
Yes, ma'am.
About the thing what we are doing is, one is we are taking material above the ground, the other we are taking material under the ground. Alright, that should be there. So in the fourth polythene bag, whatever material you will get after digging inside, all that will come into your polythene bag D. Okay. Then what you will do is, you will keep all the polythene bag individually. Okay. You will take polythene bag A and you will dry it. You will take polythene bag B and you will dry. You will take polythene bag C and you will dry. You will take polythene bag D and you will dry. Now, how are you drying? You will collect all the material individually and dry them, keeping in the oven. Okay, that is why we are using oven. We are only drying it. We are not doing anything else. So we are drying it by keeping it in the oven for 3 days. Continuously 3 days you have to dry. Okay, and how much is the temperature kept and all, that is all should be the minimum. It should not overheat, and then it should not cause any damage to the oven or to the place where you are doing this experiment. It should be to a minimum, because 3 days you are drying, so keep it to the lowest temperature and dry. You don't have to increase your temperature to 900 degree centigrade and then dry and then everything will burst up, because 3 days means you know how much it will increase, and then you should also pay electricity bill. So you should see to it you are keeping your temperatures at a lower rate. For 72 hours we have to dry them. So you imagine at what particular temperature you are placing. Okay, so for 72 hours you are drying this particular material individually. Individually you have to dry, which means it should be completely dried up. All the moisture present, be it from the material which is present above the ground or from the material which is present below the ground, all the moisture has to go. Clear? That way you have to dry. After drying, you have to remove all these and cool them. Okay? Immediately you should not cool. After it is completely dried up, you have to remove and you have to cool them. Cool, and then you have to use your weighing balance to weigh them. Okay? Understood, students?
Yes, ma'am.
Yes, ma'am.
So you are collecting the material above the ground, 3 bags you have, below the ground, 1 bag you have, or underground, 1 bag you have. After collecting, you are placing those bags separately in an oven and drying them for 3 days. That is 72-hour dry. After which you will take out the bags and you will cool those bags. When you are cooling, after cooling those bags, you will weigh those bags. So here for weighing, you are using your digital weighing balance. Okay. So how will you weigh? Again, individually, you are taking the dry weight of polythene bag A. So please mention there. Please mention observation. Okay. Write their observation in your observation books. What are you observing? So first is how much of area you have taken. How much of area you have taken to study? How much of area you have taken?
50 into 50.
50.
So 50 into 50 or 100 into 100 centimeters square. So much of area you have taken. Then the second thing is it should be dry weight of bag A or material present in A. Okay, what is the dry weight? After drying, whatever weight you get in the weighing balance, that has to be present here. Next, third one is dry weight of bag B. Whatever dry weight you are taking, it has to be mentioned here. Then dry weight of bag C. Dry weight of bag C. And then dry weight of bag D. All right. Whatever dry weight you are taking, all the dry weights you have to write. And the dry weights are coming in kgs or in grams?
In
Grams.
grams.
Gram.
Okay, they come in grams.
Grams.
So because we have taken a very small area, we have not taken the very huge field. Small area, 50 into 50 we have taken, or 100 into 100 we have taken, and from there you are separating against everything will come in grams. If you are taking the complete field, then that will be in kgs. Okay, so everything should go in grams. After which you will be doing total dry weight. Total dry weight. Dry weight of what? A, B, C, D. Total dry weight. A plus B plus C plus D. That will be equal to how many grams? Okay, total dry weight you will take. Drying in sun means 72 hours sun will come and sun will go also. You have night also in that. That is why I said you have to use very low temperature. Or if we cannot do 72 hours of drying, do not remove your material from the oven. Today you have dried for some hours and then you have switched off your oven. Ovens will definitely be hot for some time. Let them stay in the oven itself. Okay? And then, incubators will also be there in some of the colleges. You can play hot air ovens, if not incubators or your microwaves. Hot air ovens are there. Colleges have hot air ovens. which you can place them continuously for 72 hours. Okay, like how microbiology students use hot air ovens and all, those ovens are also given to the students. So you can place them and you can go home. After 3 days, you can come and take. So in incubators also, they place the material, they place the organisms, and they'll go. After 3 days, they'll come and they'll take, or after 2 days, they'll come and they'll take. So why is that we are drying it and removing the moisture, and then why are we not allowing any moisture? Is because if we dry, remove the moisture, remove the bags outside, and then we'll come to tomorrow and again we'll place. Meanwhile, what will happen is any microbial attack will happen. If any microbial attack happens, see, microbial attack means what? The moment we are out, even my hand is also having microorganism now. Your hands, though you have used your soaps, you have cleaned yourself, you are using your whatever it is called as, what is that called as, sanitizers and all that, still we have microorganisms present on our hand. Why is so? Is because everywhere in the atmosphere we have microorganisms. So microbial attack for all the aseptic things will be very soon. Now I've placed my bag inside, my polythene bag is placed inside, and then I will remove my polythene bag out, which means my whatever the moisture is there, whatever is there, it is all killed. The moment I'm removing it again, the moisture in the atmosphere is there. That moisture can again attack your polythene bag. Microbes can again attack your polythene bag. So next day when you are weighing it, there will be increase in weight. So to completely dry it off, we are not using any sun and all. We use ovens. Ovens means we usually use hot air ovens. Some places they are using the small microwave ovens are there. No? Those are also kept. Usually micro students are given these kind of ovens, either hot air ovens or small microwave ovens. So if we place it in hot air oven, and after 72 we'll make the temperature 72 hours, and then whatever temperature required, and after 3 days also you come and see nothing will happen to your material. It will be safe and secure. Okay,
Ma'am, there is one doubt. Ma'am, you are telling we need to keep the all dry materials in the polythene bag. When we keep the polythene bag in oven, it might not affect polythene bag will not damage.
that is why you are maintaining lower temperature, no? Otherwise your bag will start melting. Polythene bag is a plastic bag.
That's what,
okay,
ma'am.
So if your polythene bag is not proper, if you don't want to take that risk, you can place them into papers also. But how many papers will you place inside? Together keeping a bag is easy, or placing one large paper and spreading everywhere is easy. That might even fall. So we have 4 bases.
Yes, ma'am. But that's why there is a doubt how to keep them with.
So only I am telling we have to make lower temperatures. Your polythene bag should also not get damaged. Your bill also should not increase.
Okay.
Okay. If we otherwise, since we are taking it in grams, we are considering it in grams. Instead of taking one bag as a one bag of so much quantity, let us take small amount in a paper only, and we'll place the paper inside. Yeah, that is also easy. Here we are not taking in kgs, no. Weights we are not taking in kgs, we are taking in grams. So let us take how much ever we want. Okay, we have one full bag of material A, means let us not consider that full bag. We'll take 50 into 50 means we'll get very small amount also. You can use papers also. And then you are separating, no. You're separating everything and you're taking. So it will be very small. When we do this texture of soil experiment, that time you will actually understand. 300 grams also will be less on. You can use beakers also if you. Those things. You have alternate things. There is no need that we have to go with the procedure only. You can use any alternate things. Polythene bag is very difficult. You don't want to keep, use beakers. You don't have beakers, use papers.
Aluminum foil also we can use that what we use for the kitchen wrapping.
That only we can.
Whatever alternate methods you can do, you can use it, but the result should be, you should weigh the dry weight and give it in grams.
Okay.
Ma'am, I guess we
When
need
you are
lab
using
for
things,
this. Ma'am, I guess we need lab for this. Not at home is not possible, ma'am. I guess.
So yeah, we'll then come to lab. We'll do it in lab only.
Thank you, ma'am.
And precautionary measures should be undertaken. So see to it you will not damage yourself or the surroundings. All right, coming back to the experiment, total dry weight we are taking the dry weight in the grams. Okay, then we have to calculate with the biomass. So here only I'll write how are we calculating for the biomass. Okay, biomass is equal to total dry weight, whatever you have got of A, B, C and D, total dry weight by how much of biomass area you have taken. We have taken 50 into 50 biomass area, right? Okay. So if it is 50 into 50, 100 biomass area, or if it is 100 into 100, 200 square centimeter, that will be equal to dash grams per centimeter square. So the last answer of yours should be whatever the answer is into grams per centimeter square. Clear? This is how we are supposed to perform the experiment. Experiment is very easy. The only difficult part is by drying it off. That's all. No.
Yes, ma'am.
Drying the material is only difficult. Otherwise, everything is easy in this experiment. It is a very easy experiment. You have to only collect the materials, take the weight of dried material, and then give the biomass. So this is your experiment number 4.
Ma'am, one small doubt. Do we need to do a biomass of plant material separately and animal material separately or something like that for the above
No.
formula?
Here what we are doing, we are concentrating on ecosystem. Okay, in that 15 to
Okay,
50
okay.
area, we have plants also, we have animals also. The separation is done only by taking them separately in polythene bags. But the biomass is off together.
Okay, okay.
Okay, and this can be done if instead of doing for one quadrat, you can take 3-4 plots together. Okay, so every plot will have different biomass, means different weight. One biomass can give you less weight, the other biomass can give you more weight. So instead of doing for one quadrat, we'll do it for 2 quadrats, and we can easily compare, okay, which biomass is having more of organisms and which is having less of organisms. Yeah. Any doubts in this experiment?
No, ma'am.
Okay. So this is your experiment number 4. Please put this experiment into your record books. So from yesterday till date, I have finished 4 experiments.
Result?
Result is only so much. Okay, how will you write the result? Yeah. Standing crop of field was found to be how much? Standing crop of that particular field. Field, that's what I said. Instead of doing 1 quadrat, we'll do 2 quadrats or 3 quadrats. When we are doing 2 or 3 quadrats, we can repeat the same for 2-3 quadrats, and together we can put the standing crop. So here I have one reading. In the other 2 quadrats, I have 2 different reading. Add the reading and give a standing crop. Clear? And then biomass of the field. Biomass of the field, whatever biomass you are getting, add the biomass and give. So you should give standing crop of the field and biomass of the field. So that is one. Instead of doing 1 quadrat, please do 2 or 3 quadrats. Okay. So while performing the experiment, we'll go for 2-3 quadrats. But in the book, you write only for 1 quadrat, because again, if you take 2-3 quadrats, that will consume your pages and that will consume your time also. So in the record book, let us write only for 1 quadrant. This 15 to 50 area only. Clear?
Yes, ma'am.
Okay, we are only considering 50 into 50, 1 quadrat is more than enough because we are writing in the record book. Okay. Now the next experiment is of aquatic ecosystem. It is a very difficult to perform experiment. I am getting some disturbance, students. Please see to it you are muting yourself. Somebody called as Garata Reddy. Please mute yourself. Okay. Now the next experiment is you are seeing the same type of experiment, but you are determining in the aquatic ecosystem. So the name of the experiment, the fifth experiment name is study of abiotic and biotic components of an aquatic ecosystem. So what is the meaning of biotic and abiotic components? So you know what are biotic components and abiotic components. Biotic components are the living organisms or living factors which are present in the atmosphere, and abiotic components or abiotic factors are the non-living factors or non-living organisms present in the atmosphere. So the difference between biotic and abiotic components is very easy. You know that those components which have life are biotic components, and those components which do not have life are abiotic components. So this is a demo experiment. Write the experiment in your record books. I will give you a gist of it. There is no need for us to go in details of this experiment since we are not performing it. So what can be the biotic components when we are considering it to be as an aquatic ecosystem? Biotic components in aquatic ecosystem can be aquatic plants and aquatic organisms like fish. You have wide variety of fish present underwater. You have larvae present underwater. You have, other aquatic forms present underwater. Ecosystem again we are taking, which means we are not excluding one type of the organism. We are taking flora and fauna together inside the water. So those organisms which have life are called as biotic organisms or biotic factors. And here in that particular ecosystem, all those organisms which have life are considered. Okay. And what are abiotic components? Abiotic components are those which do not have life. Like for example, temperature, it is a factor. Then you have minerals, that is a factor. You have light, it is a factor. You have pH. pH is what? Power of hydronium ion. So why is that you are using a pH? To check the basic content, alkalinity and acidity of water. Okay. So to know the pH of water is also very much required. So here we are determining, we are not determining anything here. We are just studying. The name of the experiment is study of the biotic and abiotic factor of an ecosystem. Okay, so we are not performing an experiment. It is just a study. So you know the details. What all details are there in this particular ecosystem? 1 second, students. So when we go to the living organisms, when we are seeing the biotic factors, we have trophic levels. You know what are trophic levels? Energy levels of a food web or a food chain.
Yes, ma'am.
Okay, you have producers, you have consumers. Consumers are primary consumer, secondary consumer, tertiary consumer. So when you are placing them in a pyramid, these are all called as trophic levels. Producers are present at the first trophic level, consumers are present at the second trophic level, then secondary consumer, tertiary consumer, and as the food chain increases, you have so many consumer coming. Who are these producers? Plants are the producers. So when we consider in the environment trophic levels, you see energy keeps shifting from one trophic level to another trophic level. Energy keeps shifting. Likewise, in an ecosystem where you are considering only biotic factors, in that particular ecosystem, you have so many organisms. I mentioned some of the aquatic forms and aquatic organisms and flora and fauna. Okay, in that also you have some macrophytes, you have phytoplanktons, you have macro consumers, those organisms who are big eaters, big fishes. You have secondary consumers, you have tertiary consumers, and you have decomposers. Who are decomposers? Those who are eating, decomposing us. You have decomposers. Okay, so all of us have decomposers, be it a terrestrial organism or an aquatic organisms. Decomposers play a vital role. Why? Is because they have to liberate nutrients to the atmosphere. Minerals has to get liberated. When they are decomposing, minerals are given to the atmosphere. So decomposer also present in the aquatic ecosystem. Then okay, so all these things, you then in what do you say, abiotic components, when we are taking, how much of temperature is there underwater, at which particular region light intensity is more, at which particular region how deep is the light intensity, where it is completely dark, where there is light reaching, at what range the light is reaching. That is studied with respect to light intensity. pH of water. Why is pH of water tested? If there is biomagnification. What is the meaning of biomagnification? Do anybody know what is biomagnification?
It's the gradual increase of from primary consumer to the upper consumers, like that.
Biomagnification is the toxic content. Okay, the toxicity. Sometimes what happened, because of our activities also, the acidic content of water increases. When the acidic content of water increases, or the toxicity of the particular water increases, it will lead to the death of the organism. Okay, so the increase in toxicity is called as biomagnification. And when there is death of the organism, then it is the concentration of the organism will fall. The population of the organism will fall. And when the population of organism is falling due to the toxicity, then it is actually a negative impact on the atmosphere. Okay, we are not supposed to do such things. Sometimes because of our activities also, minerals like lead and cadmium are very toxic. They are very toxic to soil also. They are very toxic to water also. When lead and cadmium concentration increases in soil, it will affect your crops. It will affect the soil. Soil erosion takes place. Pollution in soil takes place, where you cannot plant the crops properly, and it will damage the crops. And same way, if lead and cadmium concentration increases in the water also, it is harming the organisms. So minerals are not so very, lead and cadmium are definitely poisonous minerals. Apart from that, there are other minerals. There is nitrogen requirement, hydrogen is required. Then you have magnesium required, phosphorus is required, carbon is required. So those required minerals are also required only in the according to the requirement. If you are increasing the requirement, it will create toxicity. Let us take a general example. How much ever you want, you are eating. You cannot overeat it. When you overeat it, when you are, because if we think we may not get it later, and we are overeating and overeating, we are in turn spoiling our health. It will lead to food poisoning for us. Yes or no? Overeat, something which is too much, over the requirement. It's harm. That is the nature's, a law. Nothing should be given over. Greediness also, in general, you take, if it is over, then it is actually harmful for the organism. Clear? Not everything should be to the limit. So minerals also, just because they are minerals. Okay, let us not go. If you have an aquatic, aquarium in the house, and you constantly keep putting food for those fishes, at one particular point, those fishes will die. Yes or no? Have you not experienced that?
Yes, ma'am.
Okay, only required amount of food is given to those organisms. Just because they are eating doesn't mean you put too much of food. One is the food is wasted for those fishes, and the second is most of the fishes will die. So nothing should be given over. If it is good also, it should be under limit. If you are giving too much, it is creating a toxic content in that particular area, leading to the death of the organism. Henceforth, we should avoid this. So when toxicity increases, pH of acidic content will increase, alkaline content will drop down. And if it is something like, alkalinity is also increased, basic content is also increased, and acidic content is dropping down, that is also harmful. If we take the pH of our mouth only, when you are eating too much of chocolates and too much of sweet things, acidic content increases, and then you will have problem in decaying. Tooth decay occurs. That is why God has given us a gift that is called as saliva. Salivary glands are secreting saliva to make it as neutral. So if too much of acidic content is there in your mouth, alkalinity is given by the saliva and it is neutral. Like somebody mentioned just now, balanced. Okay, so it should be balanced. That is how underwater also the aquatic organism should not be harmed due to a biomagnification process. It should be balanced. That is why we have to check the pH. If pH is too much, pH of water is acidic, then you have to put the base to bring to a neutral. If it is too much alkaline, put the acidic content to bring it to the neutral, so that the loss of population does not take place. Okay, temperature of the water is also required. Okay, so whatever the required temperature for any aquatic organism is there, that particular temperature is there. Some organisms, like for example, if you take crocodile, it is lives in water also and lives in land also. When it is too much hot for the crocodile on land, it is undergoing water, and if it is too much of water for it, it is coming on land. So it is balancing. So temperatures are also, where it's not only for us. We only consider ourselves everywhere. Temperature for us, too much of hot temperature is also harmful. Too much of cold temperature is also harmful. So we only require everything. It's not like that. Any organism in any ecosystem is a living organism, and all these light intensity, pH of water, temperature, mineral content, and then what organisms are there inside, who is benefiting whom. Okay, if aquatic organisms like fauna are benefiting from flora are benefiting from fauna, it is all in an ecosystem. They are all interdependent on each other, and they are all benefiting each other. So this is not a performing experiment, it is a study experiment. You should know about it. There is nothing to be performed, because in the experiment itself the name is given study of abiotic and biotic factors in aquatic ecosystem. Okay, if it was determination, then we should have performed the experiment. There is nothing to determine. It is the name of the experiment. It is only telling you have to study about it, know about it in detail. So it is a demo experiment. Please make a note of it. It is a very general experiment. There is no need to break your heads in this. Clear?
Are we not writing this in the record, ma'am?
You have to write in the record. That's what I'm telling.
Then
you will write in the record, but we are not performing it.
Figure and all those things we need to make.
Yes, you will be doing everything. As per in your manual guide, you will be doing everything, but we are not performing the experiment.
Okay, ma'am.
Excuse me, ma'am. Here,
Yes, Arati?
principle is given so much. We will write like introduce only abiotic components. We need pH, light, and intensity, because
See,
it's
only
too
for
much.
this experiment, listen, only for this experiment, since it is the study, either cut short the principle and write, or write only what are the important points. Clear? Don't write
Yes,
the
ma'am.
complete principle.
Yes,
But
there is
for the
too much.
other four experiments, you should write the complete principle.
Okay, fine, ma'am. Because it's too much, ma'am. We can make it small and brief.
Yeah, that's what I'm telling. Either cut short and write, or if you want to write in points, write some important points which are required. That's all. But for the other first four experiments, you have to write the complete principle. Yes, Bharati.
Can we write the definitions of the abiotic components, pH, that one?
Yeah. If you want, you write. Otherwise, if you don't want, also it's fine, because you are MSc students. You don't require those simple definitions. You already know them. Okay. Since we have to teach, I am starting from all those basics, like how we teach for PUC students. But there is no need to mention those definitions, because you all know. You can directly start with the experiment. Anything else, students? So next week, if you are coming to the regional center, please bring your complete records. Okay. I think so. I have explained 5 experiments to you all. All the 5 experiments I have explained from yesterday till today. Write all those 5 experiments in your record book.